All right, in this video, let's cover a few basic details about vaccines and how they stimulate the immune system so that we can gain long-term protection against an infectious disease without getting that disease. So, vaccinations stimulate artificial immunity, and so we want to just make sure we have a clear understanding of a couple pieces of terminology there. So first, you might run across this terminology, immunization. Immunization is a term that refers to any clinical process, so any medical procedure that produces immunity in a patient. So there are several different kinds of immunization, and vaccination is just one kind of immunization. So immunization, again, stimulates immunity. It's caused through a medical procedure. So we call that artificial immunity. If it's an active process, we call that vaccination. Whereas if it's a passive process, we call that immunotherapy. So in passive immunizations, we provide preformed antibodies to a patient. Whereas in active vaccination, we provide the antigen to a patient and they develop the long-term immunity. They learn to make the antibodies. So we really want to focus on vaccination in this video. So vaccination. Vaccination is when we expose a patient to the material that is antigenic, so the antigens of the pathogen. But that material is not pathogenic, so it cannot cause disease. And so we want to think about how do we create antigenic material that is not pathogenic, so that we can safely administer it to patients without putting them at risk of developing that disease. So, again, this is going to generate an artificial active immune response, and we want to think about how we make these vaccines. So let's look at a couple approaches to vaccine preparation. Again, we want to understand what is it that we can put into a vaccine that will stimulate immunity. All right. So we're going to prepare the antigens in some way. Again, the key to generating long-term immunity is exposure to antigens, the unique chemical identifier of a pathogen. So the first way is that we might include in our vaccine an entire bacterial cell or an entire virus that has been altered in some way, so that it is no longer pathogenic. And the first way we might alter that entire bacterial cell or entire virus is to kill it. So we know that it's actually quite easy to kill cells and viruses. You can expose them to heat or all sorts of chemicals or radiation, and then you could ensure that the bacteria, the cell, or the virus is completely inactive. So one way of preparing a vaccine is to completely kill or inactivate the cell or virus that causes the disease. And then that cell or virus will retain the antigens, the unique chemical identifiers on the outside of that cell. So here's a diagram that tries to demonstrate that process. So here we could have a bacterial cell or a virus that is capable of causing disease. We would never want to inject that into a person because they would get sick. Instead, if we treat it with heat or chemicals, we get dead, inactive cells and viruses that are no longer capable of causing disease. They cannot reproduce in the patient, so they cannot cause infection and disease. We administer that, and that stimulates immunity. But the pathogen cannot multiply, so again, it cannot cause disease. So that's the first way that we could introduce an entire cell or entire virus into a person, not make them sick, but still stimulate immunity. So there's another way. So we could also do a process to these whole cells or viruses that we call attenuation, or we make them attenuated. So some vaccines contain entire cells that have been attenuated or entire viruses that have been attenuated. And attenuation just means weakened. So it's a weaker cell or a weaker virus that can no longer cause disease. So here's a diagram that demonstrates an attenuated approach to making a vaccine. So again, if we've got a cell or a virus that can cause disease, we do something to them to eliminate virulence so that they can no longer make a person sick. These viruses and cells are still alive, but again, they cannot cause disease. So when we inject these cells or viruses into a patient, the first thing that happens is that these cells or viruses do reproduce in the body. But again, they can't cause disease. They're not harmful. So they reproduce in the body, our immune system recognizes them, and we generate long-term immunity. These attenuated vaccines, if you can find a way to safely reduce or eliminate virulence so that the cell or virus that you inject into a patient is still alive and can still replicate, these vaccines are by far the most effective vaccines. They stimulate the strongest immune response because we get this amplification of the antigen when these attenuated cells or viruses reproduce in the body. If we take a step backwards and think about the killed vaccine, these cells or viruses, when we administer them to the patient, there is no amplification of the number of antigens we're exposing the patient to. Whatever we inject is the exact amount they're exposed to. That often stimulates a weaker immune response than an attenuated vaccine where we get this amplification because of reproduction of the cell or virus that stimulates the immune system in a stronger way. So that's a really important detail to be aware of when we're trying to design a vaccine. The best case scenario is that you can make an attenuated vaccine. Now, it is not always possible to be able to eliminate virulence from a cell or virus. So it might not always be possible to make an attenuated vaccine. All right, so those are the two approaches to including the entire cell or the entire virus in your vaccine preparation. Including the entire cell or entire virus is great because you are positive that the antigens are there. But there is another approach. And the last approach is that we could identify the antigenic molecule. So what is the one chemical, or what are the few chemicals associated with that cell or virus that act as the antigens? And then we can purify just those antigenic molecules and include just those molecules in a vaccine. So here's a diagram to try to represent that. If this virus is capable of causing disease, and we know that these spike proteins on the outside of the virus are what act as the antigen, well, then we can just purify just those proteins. We could make them in a lab, or we could take apart a virus and purify just those parts, and we could use that in our vaccine. Here's a cell that might have several different chemical components that act as antigens. Again, we could kill the cell and break apart the pieces and then purify out these specific antigens, or we could synthesize these specific antigens in a lab, and then we can inject these lone antigens into a patient, and that can stimulate immunity because we've exposed the patient to the antigen. Again, this is super safe because these individual antigens are not the whole cell, they are not the whole virus. So there is no way for the cell or virus to reproduce in your body and cause tissue damage or disease. So this kind of vaccine preparation, introducing just the antigenic components, is completely safe. There's no way that that could cause disease in a person. All right, so those are all the ways that we can create a vaccine. If we inject a patient with one of these vaccines, let's consider what happens in their body. So what is the immune response to vaccination? And really, the immune response to vaccination is identical to the immune response when we get infected by a pathogenic microorganism. So the first thing that would happen in this case is we're going to administer the vaccine. The very next step is that our APCs, those antigen presenting cells, engulf that antigenic material, whether it's the entire cell or virus that's been killed, the entire cell or virus that has been weakened or attenuated, or just those antigenic molecules. The APCs engulf that stuff, they save the antigen, and they present that antigen to our T cells. So we know this stepwise process. APCs first, T cells second. Now that the T cells can recognize that antigen, some of our helper T cells use that antigen and their ability to recognize it to activate our B cells. And our B cells will learn to make antibodies that will also bind to that antigen. So we started with the APCs, we had to activate the T cells, then the T cells can activate our B cells, and now we have B cells that know how to make antibodies against the antigen. Then we know that some of those B cells will mature into memory B cells. And that is the key, right? Memory B cells are the key to the long-term protection. So once we have memory B cells, we now have long-term protection against that pathogenic microorganism. The one thing to keep in mind with a vaccine, just like when we get sick with a pathogenic microbe, this process takes time to develop immunity, right? So the reason we get sick and experience symptoms for days to weeks is because it takes that long for our immune system to ramp up this response. The exact same thing is true when we administer a vaccine. It takes days to weeks for this vaccine stimulation of our immune system to generate that long-term immunity. So we'll end there. I hope this gives you the basic details about vaccines, what they're made out of or how they're made, and then the response that triggers in our immune system. Vaccines are a somewhat controversial topic in some aspects of our society, and I hope that this knowledge of what's included in a vaccine, the basic things that are included in a vaccine, and how they stimulate our immune system can help you begin to navigate some of the concerns around vaccines, like, can the flu vaccine give me the flu? I hope you're seeing that it sure shouldn't, right? Could the chickenpox vaccine give me chickenpox? No, right? So those are basic concerns that people have. There are other concerns around the other ingredients that are included in vaccines as like preservatives and stabilizers. And there's important conversations to be had around that. But again, the main goal of a vaccine is to protect someone against a disease, ensuring that they do not get that disease. And in the end, whenever you're considering a vaccine, I hope you'll think about what are the risks of administering that vaccine. It is a medical procedure, it does involve some risk. But how does that compare to the risk of not getting the vaccine and potentially having that disease? I think that's the most important thing to be thinking about when evaluating the safety and risks of vaccines. Anyway, if you have any questions, I hope you'll let me know, and I look forward to talking to you all again very soon.